I'm very encouraged by all the talks. And very quickly, I just want to show you sort of some of the things we're working on behind the scenes, and that is in line with what we're doing. So for families, we have our FAM Find. We are going to move our gene with these publications that are coming out from the VUS column, which is a variant of uncertain significance, into the disease-causing category, and that's going to allow us to find the patients that are out there and support them and bring them into the fold. We're going to create an international registry so that we know who our patients are and we can collect information from them, and all the data collection that Terry Jo mentioned that is of utmost importance. Scientifically, we have our FAM function, continuing the basic research that's already started, our FAM BAM, which is our biomarker acceleration mission, our FAM Farm, which is all of our animal models, and FAM Bank, a biorepository, thanks to Combined Brain. And we have some samples at Stanford as well, and they're being shared between the two places, making those available to any researchers that want them. Our drug repurposing, and FAMFix, our gene therapy initiative. So that wraps up the presentation portion of this event. We ran over time on that. I apologize. But I really just want to thank all our presenters. I want to thank everybody that's been working on FAM177A1. Your work is so valuable. And to the Matts, Fuller, and Simon, thank you for showing us the future of FAM. And I invite anybody who can stay and wants to stay to stay on. And Ethan's going to lead us in a roundtable, and we can get into the weeds and get all nerdy, and or not. If families want to stay on, if you have other questions, we can answer those as well. And I'm going to be reaching out to everybody because I want to know what resonated. What do you want to know more about? I want to do more of these, but I want them to be important. I want to know what people want to dig into a little bit more. So you'll hear from me on that. But again, thank you everybody so much for coming. So Ethan.
Yeah, if you're going to stick around, please turn on your cameras. Let's try to keep it a little bit more intimate here. If I know folks, if you have to drop, we understand it is a Saturday. Thank you for your time. But if you're going to join us, please turn your cameras on. And I'll just maybe start out with anyone who had any burning question, please ask it or drop it in the chat if you prefer to do it by typing. But we can be very informal here, kind of like a breakout session. So raise your hand, I suppose, if you, okay, we can, Elizabeth, go for it.
Hi, thank you all so much for your research into this topic. I'm actually Jeff's sister. I have an open question really for anyone, but I was wondering if there's any insight about what might explain phenotypic variations within families. Like, for example, my nieces, Layla and Alyssa, they seem to display pretty big differences in severity of their symptoms. So I just wanted to see if anybody had any insight about that.
Yeah, I wanted to bring up that question too, if no one else had done it, because it's almost like in every family you see there's a mild and there's a more severe. And I've seen that pattern in metabolic diseases, like glycosylation disorders and mitochondrial disorders. So, I don't know what that means, but if anyone wants to chime in on that, please do.
I can only speculate as a geneticist that likely there might be additional genetic changes in the two siblings. So they share mutations in FAM, but maybe they have other changes in the genome that on their own don't make, don't have an impact, but together with losing FAM function, they do. We hope to learn more about this as we start looking at genetic interactions between in zebrafish between our FAM models and other genes.
Go ahead, Matthew. Go ahead, Matt. Yeah. Simon, Jack's Matt.
Sorry, my headset was not down, so it wouldn't let me unmute. I was just going to chime in off what Liliana said. It is probably a lot to do with variant genetic mutations, just polymorphism that could change function from one individual to the next. Another thing that might be worth considering, because we've seen this with a few other diseases, one of them I work on, Dravet syndrome, it has a trigger during development, which is a fever, and the fever causes the first seizures in those patients. And usually things start to go downhill for them from there. So there might be some sort of environmental cue or developmental cue that might be happening at different times, and depending how late into the development it happens could change the severity and how it presents in the patients. So as Terry was mentioning earlier, the more we get some longitudinal tracking, some life history, natural history on patients, that kind of information might help give some insight into why we see that variation.
Do you mean even like in utero, as early as in utero differential exposures or potential cryptic stresses or something, or is that less likely?
I would say less likely, but I wouldn't rule it out. The way I kind of think about it is we see that these patients, the kids develop normally. They start out, it isn't until we start seeing a few early developmental mile marks that we start to see that drop off, but they do hit some of the mile marks on time with everybody else. And figuring out when those mile markers are not occurring or when they're happening from one patient to the next might give us a better idea of when that window is, and then we can start using the other information from all the patients to figure out if there were some commonalities and why, one or a couple individuals are started in the show, more mild phenotypes, whereas some of them might have it more severe. And if there was a trigger that happened earlier in development or later in development that could give us an insight in that, it might be a tip in the right direction of what we could explore.
Should we think about formula fed versus, breastfed, how long, factors like this as early on in development? I mean, I've seen this pattern as well in mitochondrial disease where, it's like, as long, if you're still sort of being breastfed, there's like protective window there, but then as soon as that, and that always tends to coincide with 18 months, 2 years, also when potentially you start to get ambulatory or could also experience a fall or a trauma. I guess how much should we be, as long as we're on the topic of, the differences here, maybe we dig a little deep on this early development stuff.
So I wouldn't be able to say where you should draw the line. I mean, at the end of the day, all information could potentially be very valuable. It's just trying to figure out which of it we should really be tracking. And in that regard, it might be worth looking in to see if somebody can do some cross-sectional work with other diseases with similar phenotypes to see if anybody's already started tracking that type of information in other rare diseases or other diseases that present in similar ways. So again, I already recognize a lot of the phenotypes and some of the patterns happening with development in a lot of the FAM patients. With other developmental disease, there's another disease, it's called PGAP3, has very similar phenotypes, very similar presentations, and seems to have some dynamic changes with them as they go on. So looking at other groups and what they've done and what they're tracking would be a good start to figure out, what we could put into the list of things that, new families entering in could help fill out the roster of information with. And then, get a good bioinformatician at some point, start cross-referencing this stuff. That'll take time, of course. I mean, we're talking, as we see more patients with this stuff show up, it'll help round out the numbers. But, starting now and getting that information while it's fresh in everybody's head is sometimes very valuable.
Well, you say PGAP3, that's Golgi. Sure,
yep.
That was also, so there's another connection. Maybe at some point we'll go to the Golgi part of the discussion. Maybe that's on the basic.
These membrane brown proteins in the Golgi and the ER seem to have a lot of crosstalk with when they're not working functionally, what they start to present in the human beings as.
Terry Jo, you want to jump in?
Just seems like it'd be really smart to overlap with some of the disorders that have even been discussed during this meeting. So Cohen syndrome and a couple of the other FAMs, and maybe try to collect some of the same natural history data as them and look at some stuff in vitro too.
I guess I was struck in terms of variability that seemed like the infections or proneness to infection, that seemed to be kind of more like 0 to 1, where versus like something like macrocephaly was in every single case. I wonder any thoughts on that variability and drivers of that?
So like for the inflammation stuff?
Yeah.
Or the infection?
Yeah, well, yeah, I guess, I mean, I mean, information is kind of like, you know, it gets to be all kind of catch-all at some point. And, but like, just the fact that when you look at that spreadsheet and you see that there are just some phenotypes that are 100% penetrant, and then there's some that are more variable, and just thinking about, is that also just the modifiers, or can we start to think about or intuit what those modifier pathways might be based on, you know, which of these symptoms are kind of able to be toggled on or off?
I think that might be something that the cell biologists might be able
maybe
to
we
help
can get
us
into the Golgi side of things here, or just sort of thinking about like maybe even the protein itself. Like the protein is so small. Like, do we not just go hardcore biochem on it? And I mean, the Pietro's lab kind of started to do it when they knocked out Helix 1 and they showed Helix 1 was required for Golgi. They did, you know, it sounded like Helix 2 and hairpin weren't required for Golgi, but what do? A hairpin, that sounds interesting. Sounds like maybe some ligands going to that hairpin, and maybe it's and those unstructured regions, you know, that smells like a phase transition to me, that they're, you know, in one state they're unstructured, and then under some condition they become structured. That, you know, and then thinking about lipid flows, like that one phenotype in the fish where you see kind of the problem is maybe during mitosis and you get this spindle association. I mean, you know, the cell biology of like Golgi has been pretty well, you know, there's a lot of people who know a lot of that stuff. So do we like I mean, Pietro's lab is one. Do we get another person like in here to just like go to town on the protein and just chop it into pieces and do this kind of basic hardcore biochem? Because at some point, like that is going to, that's how it's going to reveal its function to itself under some, you know, number of studies, right? Or I mean, I'd love to hear, Liliana, what do you think?
No, these are great points. One question is to what extent, and that's what we are trying to understand, to what extent all the symptoms in our FAM patients are due to Golgi function, and what this Golgi function is versus some additional functions, particularly the ones that have been already published in immune system, where FAM has interactions potentially outside of Golgi. So these are some of the important questions that have been already raised. What are the target cells? What are functions of FAM in these different cells? And so all of these avenues, I think, have to be investigated. Maybe I'll also comment relating to gene therapy, when I think Matthew was talking about, you know, overexpressing a normal gene, and one has to know if the level of expression can be tolerated or not. So we have preliminary data from zebrafish that actually organism and therefore likely cells are sensitive to the level of FAM expression. And if we express FAM at higher level, higher than we used to look at intracellular protein distribution, because we have done it at very low level where we don't see any detrimental effects. But at higher level, we actually see almost like inhibitory effects on development. So that's something that we'll definitely have to keep in mind thinking about, you know, therapies.
I had a question for Matt Simon. You mentioned that, Jax, part of the scope of your work might be establishing some gene therapy strategies. Can you elaborate on that?
Yeah, so every group that we work with, and when we design these models in the mice, we design them explicitly with the idea in mind that we will be using a gene therapy or some other intervention to test these animals. So as we move through the phenotyping phase, what we're doing there, aside from characterizing the mouse so that we can put that data out there for other researchers who want to say, you know, I want to explore the cataract aspect of this disease, if there is a cataract phenotype, or if I want to look at the ambulatory issues specifically, they have that model. But at the same time, we want to use those really strong phenotypes as kind of a litmus test for when we do genetic therapy. And as Matthew Fuller alluded to earlier, probably one of the more standard approaches, what we would use for the mouse, since it's such a small gene in its cDNA form, we probably use AAV injections. And in our mice, what we typically do is we will do what's called ICV P0 injection. That's where you would put the AAV into the animal very immediately after it is born, right into the hemispheres of the brain, as we think that a lot of this has to do, especially from like the MRI stuff, with you know, brain development, but also at that very young age, the mouse's blood-brain barrier isn't fully intact, so you'll get kind of systemic viral, what they call transduction. So a lot of cells in the body will get the gene therapy. So it's kind of a broad approach, and we will see if we can actually improve those, you know, strong phenotypes and move that needle in the direction back towards correction. But right now, and this is something I was going to bring up, typically we partner with another group or another lab or organization when it comes to the gene therapy. We are starting to be able to move into a type of gene therapy called ASO, which I don't think is going to work for FAM. It's more about mRNA regulation. So if you guys don't, I mean, I imagine Matt Fuller's group might be part of that discussion, but if somebody can make it, we can test it in the animals up at Jack's.
So I had a question. I don't know enough about this disease, but I went back and looked through my notes that I took when I had a student working on it this semester. And I remembered that there is a homolog, FAM177A1, I think, or API. And I'm wondering if that could be one of the reasons why you have some kids that are a lot worse than other, if they have more breakthrough expression of that homolog, they might have it taking over the role of 177A1. And if that is the case, that you have kids doing better when they have more of that, then that's something you can exploit and turn it into a treatment. Anyway, maybe it's a total tangent, but I vaguely remembered that. And I also remembered there was an A2, and A2 also seems to perform some of the same functions. And so I would really want to look in iPSCs, compare the two phenotypes, the more severe SIB with the less severe SIB, and see if there's a difference in expression of those.
Yeah, and you could always also just go into the knockout and just overexpress the homolog or the pseudogene and just see if it can do it, compensate. Yeah, I mean, it's definitely worth testing. It's low-hanging fruit.
Yeah, and exactly, Matthew, like SMA, because if the homolog is sitting there, you could maybe, from what my student dug up, it wasn't clear that if you knock down A1, then you could get AP1 to take over. But of course, you know, if you could do that, there are several disorders right now that are in that boat and where ASOs come in really handy.
Yeah, that's what I was thinking. If this would be real, this is a nice ASO target.
Mm-hmm.
Anyone else want to jump in or feel free?
I saw, Jocelyn, you put the Metabolon link in there, and Terry Jo can speak to that. That's one of the biomarker studies that Combined Brain is doing.
Oh, nice, awesome. Yeah, I wasn't sure if I missed that, if there was any data yet for that, but we've worked with them in the past, and we do a pretty good job of, you know, having a panel of metabolites that you can read out from blood, for instance. One idea on this is more of a therapeutic strategy that I work on at Tune, and isn't yet approved in the clinic. So it has that similar caveat to like the prime editing or base editing strategies, but it's epigenetic editing. And so you can deliver a synthetic transcription factor to activate the normal copy of the gene and start to get more expression off of that allele. And so it might be kind of parallel to the traditional gene therapy strategy where you're overexpressing a new copy of the gene. And I'll share an article about it.
All right, we got our hardcore group here. So I know we got 12 more minutes left on the books. So far, this has been a great brainstorming session. Anything else, any other topics we should cover that we haven't, or we want to drill down on something else we've already covered? Now's the chance.
Hey, Ethan, it's Doug. This is a very simplistic question, but it's probably for the maths on the call. If we're able to try something, genetic engineering wise on a mouse, what does that look like? And then let's say you find a hit, and I forget all of the terminology that you use, but whether it be CRISPR or the virus or whatever, but what are the next steps after that? Like, you go from a mouse to then, how do we try to get it to a human?
So I can comment on the mouse side of things and up until we kind of finish with trials in the mice. So like I said, we start out, again, after the phenotyping phase, what I'll end up doing, hopefully I'll have 3 or 4 phenotypes using those assays I mentioned that will show, a really strong difference between what we call the litter mate control. So this is just the wild type animals that were born from the same mothers. And the animals that are showing our model. So, the double knockout. And so once I have that, we will do, like I said, those injections to see if we can correct it, probably again with an AAV, I'd imagine, and we'll just restore, expression of normal FAM within the brain and most other tissues. From that point, basically what we start looking for is initially, does that injection cause any problems for the animals? Some different systems respond differently. Sometimes that type of injection is going to cause problems. Other times it doesn't. So that's first kind of a safety test. We then will look at efficacy to see how well is the virus we're using, transforming the cells, how many of the cells actually, get the good copy that we put back in. And we look at that, and then once all that's kind of squared up, we'll use those key phenotype assays and see, are we moving that needle back towards wild type, back towards a healthy individual's place to be. And during this whole process, we do this at a level that's called like pre-IND. So basically, if the FDA or anybody wanted to come in and audit our data, we show that we use blinding on our text. They don't know what mouse is what. That's really important for a lot of early FDA stuff to show that there's no bias happening. But usually at that point, what we then is have a whole bunch of data showing that we have a good model here in the mouse. We can correct that model using an approach that would be directly translatable to the human. And we kind of turn all that data over, which basically anybody you would need somebody who can kind of help then move that into a proposal, which is an undertaking, but there's plenty of great groups out there that help with that, and put it in front of the FDA saying, look at all this great data we got. We have these efficacy studies in the mice. We've got these correction studies in the mice. And then the FDA will usually kind of work with you on figuring out, okay, where can we get this process started at, usually a little bit more down the line than people who didn't have that level of rigor put into it. But the other, Matthew might be able to take it from there, more about that process looks like, because that's kind of where my expert on the overall purpose, repurposing or intervention stuff goes to into the clinic.
Gotcha,
yeah,
that's helpful.
That was a great intro. So I take all of the other MATS data from the pre-IND and I put it into an IND, which stands for Investigational New Drug Application. And you have to be able to show that you were effective in treating a mouse or whatever model you use. Although the mouse is not required anymore, it's still the most heavily favored. You have to show safety, and you have to be able to demonstrate you have something to read out from a human. They call it a biomarker, but you have to have something clinically that can be, analyzed, assayed, read. You submit that to the FDA. They have a month to say yes or no. And then once they say yes or no, if they say yes, you move forward and you have clinical trials that they can literally start like the next day. For gene therapy, I can tell you that those costs basically that next day are $30 to $60 million to do what's called phase 1/2. You have we actually have 4 phases, but you have 3 main phases. So phase 1 has typically been safety, phase 2 is efficacy, and phase 3 is like blinded efficacy. And then phase 4 is approval. For rare disease, they allow us to put phase 1 and 2 together. And especially like for AAV, it's been done so many times. People understand what doses are typically safe and typically unsafe. And then you're just trying to get the most effective dose for therapeutic benefit. So you get the IND written from all the data that Matt gives you, get it approved by the FDA. You spend $50 to $60 million to get a trial going. You get your phase 1/2 data, which suggests that it's safe and effective to some degree. You have to go back to the FDA and say, hey, we think we have something. And they'll say, yes, you can go on to phase 3. Phase 3 will be a more in-depth study. You'll have to have more biomarkers. It's typically blinded, although there are companies pushing back against that for rare disease. And if all of that goes well, then you can get an approved drug.
Gotcha. So 50 to 60 million. So Ethan, that would be, if that was the genetic side, what about, I mean, is it all the same thing on repurposed drug, kind of the same process where we started in the animal model?
Much cheaper. Like for Maggie's Pearl, the example I showed you, the all-in costs will end up being like $5 million, so and I think a lot of the cost on the gene therapy side is the manufacturing, and then the FDA is making it problematic and making trials more expensive by requiring blinding and all these other things. When in the end, I think, we effectively showed, at least for repurposing, that an N of 1 was a phase 1/2. So they were willing to essentially accept that and we went straight from Maggie's N of 1 to a phase 3 pivotal. So I know that's only probably possible with a repurposed drug, but once you crack the door with the FDA, no modalities, I think, are not too far to follow that. So, but still, the costs are significantly less with repurposing. But you also need to have funds in the bank for, like, what if it turns out that this backup gene thesis is right? Then, there's tons of ways you could probably get a backup to go up. What if, in the case of these larger, 8, 9, 10 kb deletions, prime editing will be able to replace payloads up to 40 kb, but that's a ways away. So you need to also just think about, you might make multiple so-called genetic therapy bets, not just ones that are the classic gene replacement approach, which may be perfectly fine given how small the gene is and all that, and the fact that you're dealing actually with deletions that are out there in as the variants. So yeah, but I guess that's a separate discussion for thinking, what the decade budget needs to be if you're really talking about wiping FAM from the face of the earth and being able to do that, not just in America, but in other parts of the world too.
Gotcha. Helpful. Thank you, everyone.
Actually, if there's time, can I ask one more question? It might be a kind of a naive question, but I'm curious because I actually work in a neuroimaging genetics lab where we associate genetic variants with brain structure derived from MRI. And I was curious if there's any utility for large biobank data sets to study perhaps unaffected but carriers of the variant, and to see if there's any associations. I know it's very rare, but I just wanted to throw that out there and see if it might be useful to look at associations with perhaps just carriers of it, but they're unaffected by the disease. Or no.
Well, I think that might be very interesting to look into. Although I think where a lot of Barak's work is at right now with the cell stuff would be very informative. I mean, we know that you can have one functional copy of FAM and be just fine, And we also have heard from the zebrafish folks that overexpression can make problems for it. So how that one copy is working at the like expression and quantity level. So, you know, do we have the same amount of protein in a person who has only one functional copy? Do we have the same amount of RNA being expressed? Is the cell responding to that with dosage compensation in some way? I mean, those are questions we still need to kind of have asked, and the cell biology people, I think, would be in a very excellent place to really tackle that.
I mean, the RNA-seq data, I mean, I guess, shouldn't there be some evidence if there was if the cells were naturally trying to increase the expression, is that signature in any of the RNA-seq data? I don't know, maybe some of these paralogs are only human specific, not in the fish, but maybe there's a trace of this in that data set. I mean, it's a relatively straightforward experiment in my mind, but yeah, I mean, at the same time, I think you want to attack that problem. I think you want to just mantle the protein bit by bit the way a biochemist would. And yes, I know there's non-Golgi functions, but you know, I think you can be very systematic about trying to elucidate those, and maybe even doing it with, you know, yeah, I don't know, things that are very unbiased, like pull downs, or just things that are just going to, you know, we don't, you know, because yes, we know there's a Golgi localized population, maybe there's a non-Golgi population. I don't want to make any more assumptions, right, about what else the function could be. So we need to do pretty, you know, very unbiased experiments.
So I'm hearing a lot of suggestions for working in cells, you know, interrogating the cell more. So we need to learn from Barak what she intends to do, and in what timeline. But it sounds like we were going to put some resources. That would be a good place to put it. Is there sort of consensus around that? And like, who'd be the best? Who are the best? If it's not Barak, I mean, she has the most expertise, but she has her plate's full with her other programs. Like, who would be the best people to try to recruit to do this? You think find the Golgi experts, or?
Yeah, I put out the call to like hardcore Golgi cell biologists, and then also at the same time the chase down the inflammatory part, which may be completely Golgi independent, because I don't want to miss out on. I don't want to go all in on Golgi, and then that might be a mistake too. So yeah, but I think recruiting somebody who really knows, you know, this biology and doesn't care, doesn't matter, they don't know the gene. It would just be like any other project in their lab, like go study this gene, and someone figures it out. So to me, it's like a cell biology puzzle. And given it's only a 236 amino acid protein, I know I've worked on proteins that are gargantuous, and it's like you'd never be able to really. But now it feels like you got, you know, AlphaFold tells you what, sure, he told you what Helix 1 is doing. That's amazing. So it's like we have like part of the function's already been kind of broken down of the protein. So I think we just need to assault it in a very classical biochemical way, but also in a classical cell biology way with Golgi experts, but also the people who know that inflammatory side of the pathway, which, you know, I'm not neither, but, you know, I know we could go find those people. They exist. Just got to recruit them to FAM.
Okay.
I know time here. Any other thoughts here? I'm really grateful for the crew that has stuck around, and really you are the folks who are, you know, going to definitely be the advisors and the collaborators that will drive progress. So appreciate everyone's commitment.
I kept clarifying a bunch of stuff I said in the notes in the chat. So if you're interested in that whole homolog thing, you can copy and paste those. And
yeah, you should download the transcript of the chats for sure. I've been copying and pasting, but let's make sure Jill that gets, and Jeff that gets slurped up.
Okay. Leila, I know the transient, the zebrafish body size, that's transient. What's the significance of it being transient? Is there significance to that?
Well, you know, we think that it could be an example, I mean, equivalence of developmental delay, but it is transient. So again, I can only speculate that over time, there could be other genes that have similar function that are expressed at these later stages, which allow the zebrafish embryo to progress through development and develop into adults. And then we didn't have time to talk about this, but then there are some in older fish, we are starting to see some phenotypes that might be, because of aging and maybe inflammation. So it will be really very interesting to see what our mouse model will show. Do you see any developmental delays and so on? And if we think about the future, it will be, in my mind, as I said, it will take a village to understand function of this protein. And we would be really great to think about some collaborative grants of having mouse, having zebrafish. I think we had, there is a Drosophila model being generated and cells. So we can leverage experimental strength of the different models and also expertise of people involved, while at the same time being in communication with people who are thinking about gene therapy and biomarkers and so on. I'll just mention that in the paper that has been submitted, I mean, we already have the RNA-seq data. We intercepted integrated fish and human, and we also have early metabolomic data from both human and fish. So they can provide some clue and also a basis for designing future lines of investigation. Thanks.
And what's the best way to go after these collaborative grants? Like, what would be the next step?
As you know, Jill, we tried with Chan Zuckerberg. And if there would be another call, we will. I think there might be, we might be able to think about an NIH grant. Some institutes like NINDS Institute at NIH, they have large collaborative grants. If Matthew and others are interested, I would be happy to talk about that.
All right, maybe we can set up another call where we brainstorm grant possibilities.
Yep.
Okay.
Good idea.
Okay. Helen, nice to see you.
Nice to see you. Kids are napping, so I just wanted to pop in and say hi and listen to what the conversation's about.
Awesome. Is there anything in fish that is related to the human immune system that could be interrogated?
Of course. Actually, the fish immune system is quite conserved compared to human, and that's exactly what we would like to interrogate and try to separate potentially the Golgi function. I think the basic question here is if some of the inflammation that we are seeing in fish, is it related to Golgi function, or is it a different function? So it's not easy to dissociate these things, but we will try. But what is in this first paper, Jill, is that upregulation of immune markers is one of the common features of RNA-seq data from the children and from our mutant zebrafish.
I'm admittedly popping in at the end here, but so I apologize if my question's already been covered, but do we have any idea if the apoptosis piece is related to the inflammatory system or some other aspect of either fish or mouse or human? Do we have any insight on that?
We don't at the moment. I mean, apoptosis is also something that is popping up in zebrafish and human data. But what's the cause? We don't know.
Kind of a similar question for the hypertrophy. Is that do we know if that's in fat tissue or some other kind of tissue, and which animal model would be the best to interrogate that if it's a priority?
So we are not seeing an obvious hypertrophies in zebrafish.
Okay, sorry, I missed that piece.
Thank you, Matt. Thanks to everybody who had to go. Leila, so I think Heather Flanagan Street. in South Carolina, you know, and she works in zebrafish. Ethan and I talked to her, and she and her, particularly her husband, have a particular interest in Golgi as well. And so I'm wondering about possibly collaborating with them. And
happy to communicate.
Okay, yeah, let's set up a time for all of us to chat.
Of course.
Yeah, the paper I mentioned on our PM2 stuff, that was all Heather's lab. So that was all that zebrafish work from that was from their lab. So definitely very good scientists.
Awesome. Well, we have taken up a lot of your valuable Saturday. So I think we should probably wrap this up. Unless, Ethan, did you have any other?
No, this is great. I want to respect rest of everyone's weekend. We took lots of notes. We made lots of surface lots of insights here. Thanks. Hopefully we can turn this into maybe a group that meets on a more regular occasion, maybe monthly, a kind of FAM researcher call, community call. I've seen that work with other groups we work with. So this looks like exactly the core of the people who would show up to that. So we'll follow up if you're interested. We know everyone's got lots of meetings, but I think we just sparked a community here, and you are like the hardcore people who have stuck around. So it tells me that hopefully you'll kind of come back every month and contribute.
Thanks so much, everybody. Have a great rest of the weekend.
Good to see you, John.
Yeah. Thanks again, everybody.
Yeah.